Hi, good morning, everyone. My apologies that I couldn't be there in person to present this talk today, but I'm hoping that I can still convey all the information that's on these slides, and we'll hopefully get some time at the end to address any questions that emerge from this presentation. So I wanted to start out by spending just a couple of minutes in talking about this particular topic, which is microbial endophytes, and to at least explain what these organisms are. So essentially, they're organisms that we consider to be internalized within the plant. And we have identified some of these by growing them out on agar media, but there's a whole bunch of species that actually can't be cultured. Some of them may be pathogens, others may be organisms that for which we have no functions determined as yet. And so the whole objective of this research was to try to understand what these endophytes are, with particular regard to cannabis, and where do they actually reside within the plant itself. So we know from isolations from cannabis tissues that are surface sterilized. And so the endophytic populations are those that reside internally. And so the plant tissues have to be rigorously surface sterilized. And when you do that, we find organisms like Bovaria, shown here on your left, and Trichoderma, shown here on the bottom right, which we know from previous work are beneficial fungi. So we can assume that those are beneficial to the plant by being present in those tissues. On the other hand, we also have other fungi that we can isolate on agar media. On your left here is Fusarium, and on the right is Penicillium, and many of these can be potentially harmful or dangerous to the plant. So there appears to be a dynamic interaction between both the beneficials and the pathogenic organisms that are present within cannabis plants. Now, just quickly to summarize what the cannabis production cycle looks like, this is important because I'd like to demonstrate where we collected our samples from for this particular study. So everywhere where you see the presence of a black circle is where we collected tissue samples. So we started with stock plants or mother plants. We took cuttings from those plants and analyzed those. We looked at plants that were in the vegetative stage. We also took samples from plants that were in flower. We took samples of flowers themselves. And then last but not least, we took samples of dried flowers, flowers that came from these plants. So basically what this entire production cycle is showing you is that we went through all the steps of collecting plant material so we could look at them and analyze them for the various microbes or endophytes that are potentially present in those tissues. So here's a summary. From all of these various plant tissues, we obtained roots, we obtained stems, and we obtained leaves. We kept them separate. And as I mentioned in the previous slide, we looked at the stalk mother plants, we looked at cuttings from those plants, vegetative plants, flowering plants, as well as the flowers themselves. In addition, for a smaller subset of samples, we also sampled the cocoa fiber, which is the substrate the plants were grown in, or soil, depending on where the plant source was originating from. So we had a pretty thorough sampling procedure for this particular study. So once again, these are the stock plants or the mother plants on your left, and then the cuttings that are derived from them. And again, as the question says on the bottom, are these microbial endophytes the same in mother plants as they are in cuttings derived from them? Because these are direct descendants from the mother plant. And so we were curious to see whether they shared any microbes in common. In addition, as I mentioned, we were looking at these inflorescences of flowers. And again, the question was, are the microbial endophytes in these flowers similar to what they might be present in other parts of the plant? Is there some kind of a connection or continuity that we could draw from this particular study? So here, of course, is the greenhouse environment that I've been working in. This is where the plants were collected from. And as you can see here, these are plants that are fairly close to being harvested. And we did look at these flowers for presence of various types of endophytes. We also looked at dried flowers. So here you see an example of the flowers that are being dried. We sampled from these as well. And we wanted to see what kinds of microbial endophytes would be present in these particular tissues. So here are the research objectives. Again, the endophytic species, we focused primarily on fungi and yeasts. We had bacterial data there, but I'm not going to present that. I'm just going to talk primarily about fungi and yeasts. And so again, the questions are these microbes present at different stages of cannabis propagation? Where do they reside in this particular tissues? Are they in the roots? Are they in the leaves? Are they in the stems? And where exactly within those tissues can we find them? And then last but not least, can they be eliminated through processes such as tissue culture methods or other approaches? Because again, If you've got endophytes that are not beneficial, there have to be ways to try to eliminate them before they actually start causing problems in the plant. So as I mentioned, we did collect some samples of the growing medium. So here you see cocoa coir, which is a very common growing substrate for plants in hydroponic production. On the right, we had a few samples of soil. These were mostly from organic growers. And then again, the various stages of plants that were sampled, starting with the leaves, the stems, the roots, and going all the way from vegetative across to the flowering plants. The other thing we did, we were fortunate in that we could sample a multitude of different genotypes. And so based on the comparisons we made, we do have a genotypic, potentially a genotypic interaction with the various types of microbes. So what is the methodology that we used? We collected a total of about 60 of these samples, roots, stems, and leaves. We also had some very small samples of seeds that we threw in there just to see what kinds of comparisons we could make. The surface sterilization of these tissues involved a fairly extensive 10% bleach and 70% ethanol for at least 4 minutes, in some cases longer. That ensured that we weren't collecting microbes from the outside of the plant. The methodology that was used to analyze this was basically whole genome sequencing. And if you notice from the first slide, we had two authors from Medicinal Genomics that were part of this study, Kevin and Steve. And the data that they collected was basically summarizing the various microbial species based on the total reads or taxonomic identifications that were made from all the sequencing data that was collected. And I know that Steve and Kevin are in the audience, and so if there are follow-up questions in terms of the use of this technology and what the potential might be, I'm sure they'd be happy to answer any of the questions that you may have. So basically, we turned the DNA samples over to them, and they fed us back the information that identified all the various species and the frequencies, the abundance of all the various species in every one of those samples. And certainly one of the nice ways that Steve generated to show this data are what we call the chronoplots. And you may have seen these chronoplots a little bit earlier in the talk by Maria Rojas this morning. And hopefully this is information that you're now seen at least once before. So this is what these chronoplots look like. They're hard to see from where you are, but identified all along the outer periphery of these circles are the various genera and species of the different organisms that are found in the various samples, as well as the frequency. So in here, you've got 2%, here you've got 1%. This is the frequency amongst the total reads. Assuming the reads are 100%, then this would be the total reads. And of course, the reads from the cannabis tissue themselves were subtracted from this. So we can gather data on all of these various fungi, all of the various yeasts, as well as different bacteria that may be present in these samples. But today I'm just going to focus on the fungi and the yeasts. So when you take that data and you summarize it, what you'll see on these axes, for example, is the number of fungal reads. So for example, with the fungal reads, we're looking at anywhere from 200,000 to up to 1 million. These would be the reads that were generated from the sequencing data. And so what you see here, first of all, is 6 genotypes of cannabis. You'll see right away by looking at the size of the bars that there are differences in the total number of fungal reads that were found. So in the first 3 genotypes, there's more than there are in the next 3 genotypes. In addition, we compared vegetative plants, which is shown here in blue, and flowering plants, which is shown here in sort of a brown. And as you can see, the total fungal reads, or the total fungal species, was way more in the vegetative plants as compared to the flowering plants. And we also did notice that the fungal populations were higher in the vegetative plants and the yeast populations were higher in flowering plants. So you're starting to see some variation here in terms of genotype and in terms of whether the plants are vegetative or flowering, and in terms of whether you're looking at yeasts versus fungi. So it is a pretty complex interaction. We looked at the 2 cultivation methods, which were the cocoa coir, which is the hydroponic method, and the organic soil, as you see here on the right. Very limited number of samples of the organic soil, but overall, you do see a trend that in the organic production, there are, in general, more fungal and yeast reads or species present compared to the hydroponic. And this is not unexpected. I mean, in organic soils, you do have considerable amounts of microflora, and this is starting to show in terms of the percentage that are colonizing the various tissues. And we've seen this before when we've compared organic with hydroponic production. There's more microbes present in the organic. When we look at genotypes again, as I mentioned in the beginning, there are variations in the levels of these fungal reads in the various tissues. These were single samples for DNA analysis, and so you don't see error bars or any statistical separation, but you do see trends. You do see trends in that some genotypes have apparently more fungal reads than others. And here again, we compared inflorescences that were fresh, which had not been dried, compared to those which had undergone a drying process. And as you see in this slide, and this is to be expected, the drying process does reduce the total numbers of fungal reads that one can collect or isolate from these tissues. And so drying definitely has a detrimental effect on the survival of these various fungal species. And that is obviously one of the reasons why we do dry cannabis samples, is to allow the microbes to not persist. But you will see there's still a low background level of these microbes. So here's just some data to show you the various types of genera that are found in various genotypes. And I just want to highlight a couple. Here in this particular genotype B, the dark blue is Alternaria. We did see a very high frequency of Alternaria species. And in this genotype here on the right, the light blue, we saw a very high incidence of Fusarium. The dark brown bars here is trichoderma. These growers were using trichoderma in their production, and so it's not unexpected we would see high levels of trichoderma. So we are seeing differences in genotypes, not just in the total reads, but also in the specific genera. So a little bit more Alternaria here, a little bit more Fusarium there, some trichoderma over here, and the rest of the species are at a fairly low level, including things like Aspergillus, which was very, very low in these particular samples. So this is a fresh inflorescences. And then when you compare that to dried, again, let me just go back and show you that the scale bars here are from 1,000 up to 8,000. Obviously, when you dry this, the numbers go down. And so on this reads, the numbers are from 200 to 1,200. So even though the bars may look like the same, there's actually way fewer because they were dried. So here in the dried samples, again, the orange bars that you see here is penicillium. So we're seeing a lot of penicillium surviving the drying process and still showing up in these samples. The green bars here is the Rhizophagus, which is a mycorrhizal species that I'll come back to later. That appears to survive or be present despite of the fact that the flowers were being dried. So there's some interesting species or genera that we can see showing up in these various samples. Again, Aspergillus, which is shown here in the sort of a dull red, is quite low in most of these samples that we analyzed. So here's an overall summary, and this might look quite complicated initially when you first look at it. But what we've basically done is we've said, all right, here's the mother plant. These are the cuttings. This is the vegetative plant, flowering plant, and seed across the top. And R is root, S is stem, and L is leaf. Okay. And then as you go down, as you look on the left-hand column, these are the various genera that we identified listed in alphabetical order. So as you work yourself across from left to the right, these arrows are basically showing you the presence of that particular genus across all samples. So when you look at something like Alternaria, you'll see that it's present in the mother plant, in the cuttings, in the vegetative plant, flowering plant, all the way across to the seeds. Similarly, Aspergillus all the way across. So the arrows are showing you there appears to be a trend going from left to right of the presence of these various species that are found in the samples that we looked at. The lines that you see that are highlighted in either green or yellow are basically telling you that these are the most abundant. So when you look at Fusarium, for example, it's highly abundant. The numbers that you see here represent the percent of total reads. Fusarium is found in every single part of the plant, in every stage of sampling that we looked at, including seeds. And Fusarium oxysporum was one of the prevalent species. Similarly, penicillium is found in every tissue sample from every different type of plant source that we looked at, including seeds. And then Rhizophagus, which is a mycorrhizal fungus, quite a bit of a surprise to find that this particular genus was present in every tissue sample all the way across the different plant types, including seeds. Now, one thing to note is that the cocoa fiber medium was a source of many of these microbes. So, for example, Aspergillus, Fusarium, Penicillium, and the Rhizophagus were present in the cocoa fiber medium, suggesting that this potentially could be one of the sources of these various microbes that are found. Then what apparently they must be doing is getting into the mother plants, and then from the mother plants, they're transmitted over into the cuttings, the vegetative plants, flowering plants, and seeds. Our seed sample was quite small. I think we only had 3 samples of seeds. But again, it's interesting to note that there are these species that we would normally find in seeds, such as Fusarium and Penicillium and Alternaria. But also this one was a bit of a surprise that you'd find a mycorrhizal genus present in the seeds of cannabis. And similarly for yeast, not a whole lot of yeast species were present. Candida was the most prevalent. And again, it was found all the way across, starting from mother plants, cuttings, and all the way to flowering plants. And then Saccharomyces, which is a very common yeast, was also present in these samples. So much less frequency of yeasts compared to fungi. So here's an example of the Penicillium species on your left, and of course the Rhizophagus species on the right, which produces these large spores. This is a beneficial fungus. This is a mycorrhizal species that has been previously reported to occur in cannabis. Over on the left, this is a Penicillium chrysogenum, which is the species that we found, which is a very strong antibiotic producer. And we think, at least initially, that these two species are probably beneficial. The Penicillium perhaps producing antibiotics that would prevent the growth of other fungi, and then the Rhizophagus providing some opportunities for the plant to perhaps grow better in the presence of the mycorrhizae, because they were found in each and every one of the samples that we looked at. Now, the next question is, where are these microbes found? And so I'm going to walk you through a series of experiments that we did using scanning electron microscopy, starting with the mother plant. So here's a cross-section of a mother plant. If you've never actually cut one open, you'll see there's a little central pith here in the middle. And then this tissue here is the xylem tissue that you'll see over on the right with the scanning microscope. And then on the outside here is the phloem, and then you've got the epidermis on the extreme outside. So going from left to right, there's your little opening, the little pith that you see in the middle. And then these are all various types of cells progressing all the way to the xylem tissues. The reason I mention all of this is because it becomes really important as we walk through these particular slides to try to determine exactly where these microbes are hiding out. Now, this is a cross-section of a cutting. This is the cutting that's made from the mother plant. And again, very similar, except the pith opening here is quite large, as you see. You've got the pith cells, the pith parenchyma cells on the outside, as you see here, same as the mother plant. And then you've got the xylem tissues here lining up outside the actual parenchyma cells. So where are the microbes? Okay, here's a close-up of the pith parenchyma cells. These are the cells that surround the actual pith opening. You can see the intricate manner in which the cells are formed. And down below here is the xylem. This is the water-conducting tissues. This is the tissues that the plant uses to move water and nutrients up and down the plant. On your left is a close-up of these pith parenchyma cells. These are the cells that surround that pith opening. And over on the right are the xylem vessels. The xylem vessels have 2 cell types. One is called the vessel elements, which are these large openings here. And then the tracheids are these smaller cells that you see lining on the other side. So these large cells here are going to be very interesting, as you'll see in a few minutes, which are the vessel elements. So here's a close-up of the xylem tissues. If you've never seen the water-conducting tissues in a plant, this is a really good image of it. There is some material here, most likely water, that's found inside the vessel elements. And then these are all the tracheid cells that are found on either side. This is very, very common in all plant species that have this vascular system. Here's again the pith parenchyma cells. These are living cells, so they do have cytoplasm, as you see, very beautifully organized within the plant. And they also have a function for not just transport, but also storage of nutrients in the plant itself. Now, these interesting coil structures are the xylem fiber cells. So these are the xylem fibers. This is what gives plants, including cannabis, their upright, strong stalks. And you can see there's sort of a circular winding pattern, which gives them considerable strength. And over to the right here is one of these vessel elements that we'll spend a little bit of time looking at in more detail. So here's a close-up of the vessel elements. And as you get in close, you'll see there's little pores. We call them piths, so they're called piths in the literature. And these are long tubular-like structures. And a lot of the xylem material, the water and the nutrients, flows in these particular cellular components. Here's a close-up of these holes or pits, as they're called. They're about 5 to maybe 10 to 12 microns across, so fairly small. Some of them can be a bit larger, up to 15 microns in diameter. And they also facilitate the flow of water and nutrients, in some cases moving from sideways as well as up in the plant. So very interesting cell type. And here's just a close-up that shows where one of these walls is broken. And you can see in the background these various pits or openings that are present in the xylem tissues. Now, where do we find these microbes or the spores? So these are now the vessel elements again. You can recognize them with these little pits in the background. And look at these spores. There's spores of various sizes. There's spores of various numbers, all congregated somewhere sticking to the inside walls of the xylem tissues. Okay, so we opened up the xylem tissues and we looked inside with our scanning microscope and we found them. We found these spores. There's all types in here. This is a close-up. You can tell there's got to be at least 3 or 4, if not 5 different species based on the size and the shape of these particular spores. They seem to be stuck inside this xylem tissues, at least for the purposes of the microscope. This is where we actually see them. Here's another view of these spores. We know they're spores, and they're stuck to the inside of these various xylem vessels. Here's some more close-ups. Different types. You can tell this fungus is different from this one over here. Different species. We can't identify them just based on size or shape, but we obviously know that they are fungal spores. So in terms of the actual pit openings, there's some spores, as this one shown here, that probably aren't going to make it through here. They're way too large. So the only movement they'll be doing is up and down in these vessel elements. However, there's others like this one that's small enough that it can pass through these pit openings, and so they could be movement of these spores sideways in addition to up and down in the xylem vessels. Here's a close-up. These spores seem to be surrounded by some kind of a matrix, some biological film that's present, probably with the material that's also found in the xylem tissues. But again, a first-time view of the inside of a xylem cell showing you spores of potential microbes or endophytes. So the other question I wanted to ask is, we know there's spores moving up and down in the stem of the plant, in that case, the mother plant. What about in the flowers themselves? So this is the inflorescence tissue. So I've decided I was going to go in here and with a scalpel just cut this stem and take a close look at it in the microscope. Okay, so here's a close-up. This is the actual flower bud itself, and this is the section that was made to reveal what was actually potentially present inside the tissues. So when we did that with the scanning microscope, lo and behold, not only did we see spores, we saw lots and lots of spores inside those little stems that are inside the flowering tissues. So much so that I've never actually seen this many spores hiding out inside the xylem tissues in the inflorescences themselves. This is obviously not a good thing because you don't want these spores to be inside your flower tissue, but there they are. And in fact, they're found in very high numbers, as you can see in this particular micrograph. So different types of spore sizes, different shapes, and there appears to be an abundance of these spore types in that particular tissue that's from the inflorescence stem. In some cases, we actually saw mycelium. So you see mycelium here growing across one of these pit parenchyma cells, and I'll show you a close-up of that. It almost looks like a spider's web, clearly, but it's mycelium. It's mycelium that's growing from the outside of the actual fungus itself. So very, very interesting to be able to see this close-up of what's going on inside the vascular tissue of a cannabis plant. Okay, so here's the summary. We did find these spores in stem tissues, in inflorescence tissues. We found them in the pith parenchyma cells. We found them in the xylem vessels. We think the spores are being moved around with the water and the nutrients up and down the plant. And we also think that some of the sources of these microbes is the material, the growing medium, whether it's cocoa or soil, that might be allowing these microbes to gain access to the plant. Now, before I end, I just want to address with a few slides what tissue culture can possibly do to eliminate the endophytes. And I'm building on Maria Rojas's presentation earlier this morning, where she showed convincingly that when you do meristem culture, you can eliminate many of these microbes because of the fact that you're using such a tiny piece. When you use cuttings or nodal cultures, you don't find elimination of the microbes. So meristem culture will eliminate many of these. Okay, but the question we wanted to ask is what happens to those tissue culture derived plants? What we found coincidentally was that many of these tissue cultured plants were very, very easily colonized by things like Fusarium, pathogenic Fusarium. And they were also very easily colonized by beneficial fungi such as Trichoderma if they were present in the environment. So what we're thinking here is that when you eliminate the resident microbes in a cannabis plant, you're potentially exposing it to infection by a pathogen like Fusarium, or you're potentially allowing other beneficials to colonize and grow through the tissues. So let me show you the chronoplots that came from a tissue cultured plant that was exposed to Fusarium. You can see with the red outline that almost 90% of all of the DNA in that particular plant was Fusarium. It was actually colonizing to the extent that the plant actually died. Okay, you compare that now to a plant that received a trichoderma. We actually artificially added trichoderma, and you can see that the trichoderma has colonized again. Almost 50% of the DNA is now the trichoderma. So this is a bad story, good story ending. The bad story being tissue cultured plants may be most sensitive to Fusarium. On the other hand, they're very receptive to being colonized by trichoderma, which is a beneficial, and that would allow it to establish itself within the plant. Okay, take-home messages. We find a lot of very interesting microbes. They were present in mother plants, in cuttings. We think that the endophytes are influenced by genotype. The two that we found most commonly were Penicillium and Rhizophagus, which is a mycorrhizal. And in the inflorescences, surprisingly enough, we did see quite an abundance of spores of these various fungi. And they were also found in the xylem vessels and in the pith parenchyma. So basically, the summary here is the vascular tissues of cannabis plants appear to provide an avenue by which endophytes can be transmitted internally and potentially reach the inflorescences and the seeds. And there have been a number of studies that have shown seeds do indeed have a large number of endophytes, and possibly they originated through the xylem tissues. So here's the idea then, a summary. These arrows pointing upwards show the progression of endophytes within the plant, potentially moving all the way up into the inflorescence and colonizing those tissues. And this is a final summary. Basically tells you that endophytes were found in all of these various plant tissues. They may be transmitted from plant to plant. They may be transmitted by water. They may be transmitted by people working with these plants. We still don't know exactly the sources of these various endophytes, but they are there. And they're there in some cases in very large numbers. So with that, I'll conclude with acknowledgement of the funding sources and the various people that have contributed to the research and have helped us get as far as we have. And I look forward to hopefully hearing questions from the audience and hopefully being able to answer them. So thank you very much again for listening.
All right, first question. Hello, Dr. Punja. This is Soren. Thanks for sharing all this important information. You showed that alternaria is in the inflorescence tissue, and that was very interesting because we know alternaria to be a localized, foliar disease. And if you have found pathogenic species like alternata, I think that this is the first report showing that it can be a vascular systemic disease. Thank you. Do you confirm that?
Oh, yeah, thanks, Soren. We've actually found alternaria on inflorescences, and there are reports of it being actually on the seed as well. It's also known as an endophyte in certain plant species. So there is a likelihood that it could enter through various avenues and find its way into the xylem tissues and work its way up into the inflorescences and into the seeds. Remember, too, that it's also airborne. So there's always opportunities for alternaria to invade not just the leaves and stems, but also the flowers through airborne mechanisms.
Hi, Zamir. This is Mike DeHolis. Great presentation. Two quick questions. One is the spores in the vascular system, those are the result of sporulation within the plant. Is that the idea,
Yeah,
right?
yeah, correct. Well, sporulation and potentially movement through the plant. I mean, you know, and I know that there are vascular infecting fungi like Fusarium and Verticillium that are able to sporulate within the xylem tissues themselves. It's interesting that the endophytes can do that and presumably build up their populations and move through the xylem vessels. But up until now, of course, we haven't been able to isolate and culture them. But that would probably be the next step.
Right. And then second question. So the susceptibility after meristem culture to Fusarium, is that specific to Fusarium or did you test other fungi as well?
That's a super question. I mean, the Fusarium contamination was accidental. Obviously, we didn't want the Fusarium to be present in the tissue culture room where these plantlets were being grown. Somehow it made its way in there, and the plant started dying. And so I quickly ran off and sent a couple of samples to medicinal genomics, and they gave me the results back that 90% of the DNA was just Fusarium. So it kind of begs the question: were those endophytes previously there, providing some beneficial unknown properties such as protection against these invaders? We haven't looked at for other fungi. Fusarium seems to be the main one that invades into these plants, but presumably other pathogens could also gain access in the absence of this in the presence of this microbial void. So that's a really interesting idea. And I know that your lab and your students have shown that seed obviously is a carrier for many of these endophytes, which to me makes total sense that they gained access presumably through the xylem tissues, got into the seed. And then when you plant that seed, it provides the immediate, presumably, protection against these other invaders.
Great, thanks.
Hi, Dr. Punja. Mike Catalano here. In the plants that had spores in the, high concentration of spores in the xylem, is there any evidence of mechanical interference with water or nutrient movement?
Good question. I don't know. One would presume not, but then, just the physical act of having these spores and mycelium in the plant could provide some negative consequences. I mean, we've always assumed endophytes are great. They're wonderful to have because they're in the plant, they're there for a purpose. But they could also be having negative aspects to the plant. One could be the reduced translocation of water and nutrients. Maybe the plants are not growing as well as they should. And we know that they are detrimental endophytes. And of course, Fusarium, again, is the one example. So again, remember, the scanning microscope amplifies everything and makes it look like the world is coming to an end with spores. But in the relative sphere of things, only a few cells were colonized that we could find. The rest of the plant seemed pretty okay. So it may hinder water transport, but I'm not sure.
Hi, Dr. Punja, Darren Kaplan here. So you've shown that the plants will readily take up inoculants after tissue culture, after meristem culture. I'm wondering if you've thought of any implications for microbial release testing for dry flower and how you might be able to inoculate something that might not show up for total yeast and mold, for example, in the finished product.
Yeah, that's a good. Well, so there's two answers to that. The finding of enhanced colonization right after tissue culture is super important. We can't let those tissue culture plants out until we've inoculated them with something beneficial, particularly if they've originated from meristems, because you're asking for trouble. A good portion of those will definitely make it up into the flower. We know that things like Trichoderma and Penicillium, if they're added to the root zone, will eventually end up in the flower tissues. The good news with the drying process, though, and this is some data that came very recently, is Trichoderma does not survive drying. And so you can add as much of that particular biocontrol as you want, but the drying process actually kills the spores off. Penicillium is a different story. That one's quite resilient to drying. And so if you were to add penicillium as an additive, for example, Penicillium chrysogenum, which we think is a beneficial species, you may end up failing some of these yeast and mold tests because you've increased the populations to a level where even the drying doesn't reduce populations. Now, one other study that we did very briefly was that when we added a systemic fungicide, and of course, there are no fungicides registered, But on an experimental basis, when we added a systemic fungicide, it completely cleared out all of the endophytes. They were completely killed off in addition to all the pathogens. So I'm not recommending we do fungicide testing, but clearly there are ways that good and bad endophytes can be eliminated with a fungicide application.
That's great. Thank you.
Any other questions? All right, no other questions. Let's give it one more time for Dr. Punja.
Well, thanks a lot, everyone. I'm sorry I couldn't be there. Personal circumstances prevented, but thank you.